Okay, so you've been studying a bit bacteria and viruses with your pathogens project, and bacteria were part of the kingdoms project, so you know a little bit about them, but I want to review some things here that we need to know still. So with the group Prokaryotes, now notice I have it labeled bacteria, and that's sometimes just sort of a term that can apply to prokaryotes, but as you all know, that prokaryotes are divided up into two groups. Here's just a list of some different bacteria, and some of these names like Staphylococcus and E. coli and Streptococcus are probably familiar. Salmonella. These are some bacteria that in some cases cause some problems for us. All of these, though, are not bad. These two guys right here, Nitrobacter and Rhizobium, are very important in terms of the nitrogen cycle. Lactobacillus is important with making yogurt and things like that from milk. But yeah, some of these are things you want to avoid, that's for sure. Yep. But when we look at prokaryotes, you all know that they're divided amongst what we really call the Eubacteria and the Archaea, or the Archaebacteria. And both these together are prokaryotes. And then the rest of living things are all eukaryotes: protists, fungi, plants, and animals. And again, these prokaryotes are divided up in these two main groups. They presumably have a common ancestor to each other. And so prokaryotes have a pretty basic structure. They are, of course, made of cells, and so they have a cell membrane, plasma membrane, excuse me. They often also have a cell wall, and they sometimes have a capsule, this sort of sticky material that surrounds them that will have these projections called pili, and that capsule sort of helps hold them in place. The cell wall helps them maintain their shape, and of course the cell membrane is a semi-permeable membrane that controls what goes in and out of the cell. Some of them have a flagella for moving, some of them can move. Inside, they're pretty simple. They have some DNA and ribosomes and proteins and such, but you'll recall they don't have chloroplast or mitochondria or endoplasmic reticulum, those kinds of things. Now, they still need to do all the things that other cells do, cellular respiration, for example, and some of them do photosynthesis. They just don't have those specialized organelle for doing that. Okay, let's focus on the archaea. These interesting ones that are found in unique environments. There's a group known as the methanogens that you often find in the sediments of swamps, and their claim to fame is that one of the waste products they produce is methane gas, CH4. They live in these relatively anoxic or oxygen-free sediments of swamps. You also find them in the stomachs of cows. I don't know if you know, but cows have a 4-chamber stomach, and one of those stomach chambers is full of bacteria and other microorganisms that help with digestion. It also means that cows can produce a lot of methane in their flatulence. You also have some archaea that are what are known as extreme halophiles. They live in very salty conditions. Here we have these ponds where they have taken seawater and they're letting it evaporate to produce, what's the white stuff? Sea salt. And so the different colors you see in these ponds are different types of Archaebacteria that are thriving under those really salty conditions. A place like the Great Salt Lake out in Utah will have some of these organisms, as well as the Dead Sea. You have some that live in really hot conditions and also very acidic conditions, the thermoacidophiles. So these hot springs like you see out at Yellowstone, almost to the boiling point, these bacteria can live there and again thrive there. Deep in the ocean you have these vents, these thermal vents where there's this hot water streaming out of the seafloor. You'll find some of these funky bacteria living there, as well as these unusual animals, these tube worms, for example. They're deep in the ocean. Okay, now just the Eubacteria, what we sometimes just call the bacteria, the other group of prokaryotes. A wide variety of metabolic processes, great diversity. 3 basic shapes with these. Some are spherical, or they have the cook-eye shape. Some are rod-shaped, they're bacillus. And some are spiral-shaped, or helical. Sometimes you'll see that as part of their name, like Staphylococcus. It's basically a staph that has this round shape. The bacteria also have some unique cell wall structures. Some of them have a very thin cell wall with 2 membranes, and others have a thick cell wall with a single membrane, and these guys with the thick cell wall are known as gram-positives, and the rest are known as gram-negatives. Because of their thick cell wall, gram-positives retain a lot of this, what's called gram stain, and the gram stain technique is used to differentiate these two types. And so gram-positive bacteria like anthrax here show up really dark purple when you stain them with this technique, whereas the gram-negatives do not show up dark purple. Gram-positives on average tend to be a little more on the pathogenic side, but not completely. Okay, another group of bacteria that are very beneficial for natural systems, cyanobacteria. These are the photosynthetic bacteria. You find them obviously in aquatic systems, in saltwater marine systems, and freshwater systems. You can see these guys are forming these long chains of cells, but each of those cells is basically there to do the same thing, to photosynthesize. They're a significant part of the plankton of aquatic systems. They have these interesting cells called heterocysts, which allow them to take nitrogen out of the air, which is very useful for them to get the nutrients they need, the nitrogen that they need. And there's some of those spiral-shaped ones, the spirochetes, again, a particular group of bacteria. Up close, one of these guys. There's, again, streptococcus, so those ball-shaped ones that can cause strep throat, gram-positive. Lactobacillus, bacillus-shaped, these rod-shaped ones. And this one, lactose is milk sugar, and so this is a bacterium that loves to consume milk sugar and is used again in making yogurt. You have some bacteria that are fungus-like, these long thread-like cells, and they're decomposers you'll find in the soils of forests and other natural systems. E. coli, you've probably heard of E. coli. Here's the whole name, Escherichia coli. Again, there's that scientific name, that two-part scientific name in italics, second part not capitalized. Now, E. coli are what are known as an enteric bacteria. They live inside of animals. We have bacteria, E. coli, that live in our gut, and cows have them, and chickens and pigs and lots of other animals. And we have the kind we typically find in us are quite beneficial for us and help with digestion. But if you happen to eat something that has, say, E. coli from a cow or a pig, your system's not used to that kind, and it can cause food poisoning. All right. Now, bacteria, again, like I said, have many different types of metabolic processes, and not in any one bacteria, but in the whole group, because you have some that are heterotrophs, they have to consume things, some that are autotrophs. You have some like the cyanobacteria that are photoautotrophs, they make use of sunlight to produce their own food. But then you have some like the chemoautotrophs that use molecules as a source of energy to make their own food. So they're an autotroph, but they do not use sunlight. Okay. Some of them, like anthrax, for example, can form these things called endospores, which is a dormant, dry, resistant form of the cell that can just sit and wait and wait and wait in very dry conditions until it gets wet again, and then it'll wake up and start doing its thing. And so anthrax is, when it's used as a weapon, if you will, is in its dry form, and if someone were to inhale it into their relatively wet, warm lungs, then it'll wake up and start causing problems for them. Again, a lot of them have flagella for swimming. Now, when we studied genetics, we talked about these things called plasmids, which are extra pieces of DNA that bacteria sometimes have. And you remember those plasmids can be moved around from one bacterium to another, which is useful for moving DNA from one to another. Many bacteria, many beneficial bacteria in particular, are what are known as saprophytes, and they're bacteria that are decomposers. They're found in the soils of forests and all natural systems, and when leaves and sticks and whole trees fall down, these bacteria start eating that dead material. A very beneficial activity to help recycle these materials and get them back into the system out of these dead organisms. And things like the cyanobacteria, again, are known as photoautotrophs. They make their own food using sunlight. and a significant component of the plankton and aquatic systems. And again, there's some of those chemoautotrophs. Now, where do you think we're going to find those guys? Since they make their own food, but they don't use sunlight, you're going to find them where there's no sunlight. So deeper in the ocean, sediments of swamps, perhaps even in caves. Now, some other unique things. An obligate anaerobe. Now, if something is anaerobic, what does that mean? It means it does not use oxygen. If you're an obligate anaerobe, it means you do not even like oxygen. In fact, oxygen is toxic for you. So there are some bacteria that cannot handle having oxygen around. And again, they're found in oxygen-free environments like the sediments of swamps, sometimes in the stomachs of cows. Also, this bad actor right here, Clostridium botulinum. This is a bacterium that thrives in foods that were not properly sterilized and canned. And so under those relatively anoxic conditions inside those food containers, they can thrive, and this is a significant type of food poisoning that can kill people. Facultative anaerobes are organisms that do not use oxygen, but they don't care if it's around. E. coli is an example of this. They don't use oxygen, but if it's around, it's not a big deal. And then others are obligate aerobes. They have to have oxygen around. We would be an obligate aerobe, and there are also bacteria that are like this. Look at this guy, Mycobacterium tuberculosis. Tuberculosis, it's an infection of the lungs, and as you can imagine, there's lots of oxygen in your lungs, and so these guys need that oxygen to do their thing. Okay, now we talked about transformation. This is just to refresh your memory from the genetics section, how you'll remember bacteria can take up pieces of DNA from their surroundings, from other bacteria that have fallen apart or died. Conjugation is another way where two bacteria get together and they form a connection, and DNA moves from one to the other. Don't worry about these terms right here, F plus, S minus, F minus. Just know that conjugation is when two get together and DNA moves from one to the other. And transduction is when DNA gets moved from one bacterium to another, but it's via a vector, and this vector will be a virus. This virus literally picks up DNA from one and transfer it to another. All these techniques are ways that bacteria can acquire new DNA and new genes, because as you know, bacteria just reproduce asexually and make clones of themselves, but these processes can bring genetic variation to them. Okay, many bacteria are beneficial. These decomposing bacteria out there, the ones that take nitrogen out of the air, but there are some that are pathogens. They cause disease, and so people who are pathologists are doctors who study these diseases. Sometimes they're problems and pathogenic because of toxins they release, exotoxins. Other times they're problems because of endotoxins. These are materials that are inside the cell, and when the cell is damaged and breaks apart, it releases those toxins. And so either way, whether it's on the outside and just released from it or released from the inside, they are materials that cause problems in us and give us the symptoms of our illnesses. How do we fight them? Of course, with antibiotics. Compounds that are anti-living. They kill living things. And of course, you want them to focus on the bacteria and obviously not kill you. Now, antibiotics are amazing things and have helped human welfare. But what can happen to the bacteria? They over time can develop a resistance. So the bacterial population can evolve over time to become resistant to certain antibiotics. So you shouldn't say a person becomes resistant to the antibiotic. It's not the person, it's the bacteria that are residing inside of them that have evolved and become resistant. So ciprofloxacin is an antibiotic, and you can see here this change in time of the bacterial colonies becoming more and more resistant to it. Of course, we have to be careful with the foods we eat, make sure we clean them, we clean our hands before we eat so that we don't transfer bacteria like the bad forms of E. coli, that we store it properly in a refrigerator so it doesn't rot, and we cook it properly. Like meat, raw hamburger has to be cooked to a certain temperature to kill any kind of bacteria that are in it so that you don't get sick and have food poisoning. Okay, that's it. Next video will be viruses.